After we have produced the design matrix, then the actual analysis should be performed. And we know that the Lima has the program that is known as the empirical Bayes. So it uses the method that is contains the certain parameters, and that is why it is called as the empirical. And Bayes is described certain approaches that are used in the statistics. So that's why it is known as the empirical Bayes. So this empirical Bayes method is much more better than we are using than the different statistical methods like the t-test and the other tests. So we can use the command IMFIT and then quotes. And by using this command, we have to use the second command, that is EBAYES, eBayes, and then the quotes. By using the IMFIT, we can perform the data matrix, and then we can produce the design matrix. And this Lima empirical Bayes package can be used for both the filtered and the unfiltered data. For the unfiltered data, we can use this command, and we use the data matrix. This is dat.m, and we then use the second command, that is eBayes. So we use the two commands, this one IMFIT and then eBayes. And for the filter data, we again use the same commands, IMFIT and eBayes, but the only difference is that here we use the data.m and here we use the .f. So the only difference is that of the data matrix that we are using. So the object fit contains the results of the analysis. Results can be extracted using the command, that is top table and then quotes. By default, it gives output for the first column of the design matrix. However, this coefficient is seldom of interest, but the coefficient to report can be changed in the function call using the argument coefficient. If the experiment was a comparison between two groups, it could be control and the treated. The following command would extract the genes that are statistically significantly differentially expressed between the control and the treated groups. And again we use the command top table, and we use the arguments fit and then coefficient is equal to 2. And then when use the command, we will get the data in this form. And there are several columns and there is a data in the columns. So the first column is, we can see here, this first column, that is without any name on the top of the column. So this first column is the row number in the original data. It means that this is 1437 is the actual number that is present in the row. This is 527. It means this is the actual number in the row, and so on. So in this case, the most significant gene was the one on the row 1437, because this is on the top. So it means this is the most significantly has the expression for this gene of the data matrix, so that we have given to the IMFIT. So the next row is the log FC, and this reports a log 2 based fold change between the groups. So this is the fold change that it shows for each of the gene. So we can see here that some values, they are negative and some are positive. So if the value is negative, it indicates the down regulation. It means that the negative value means that the expression of the treated sample is much lower as compared to the control. So if the value is positive, it would indicate the upregulation in the treatment group. So it means that if the value is positive, then the treated sample has the higher expression level of certain gene as compared to control. So there are two statistics, T and B. Here we can see T and B. So the T is the moderated T statistics, this one, from the empirical based method, and B is the log odds that the gene is differentially expressed. The two p-values column contain the raw p-value, this one, that is generally used to indicate the expression of the gene, and the p-value that is corrected for the multiple comparisons using the Benjamini and the Hochberg's false discovery rate. So by using this different columns, we can analyze the data after normalization and quality check. So after analyzing the data, we can clearly see that some genes are upregulated and some genes are downregulated.